In this video, we're going to talk about statistical study stages, collecting data. So when doing experiments, we have two types of errors. One is called sampling error, which is basically caused by natural variation between samples. So when we select a subgroup from a population that we study, that subgroup will be slightly different than each time. It is unavoidable, this error. So a non-sampling error could be due to various human errors, non-response, poor study design, biased methods, etc. So because we cannot avoid sampling errors due to the nature of the data, we will try to minimize non-sampling errors as much as possible. So here are some examples and try to see if this is a sampling or non-sampling error. Example 1. Prior to the 2016 presidential elections, Bobbert surveyed 10 random people, of which 4 said they would vote for Trump and 6 said they will vote for Clinton. The actual popular votes was 46.1% for Trump and 48.2% for Clinton. So was it sampling error or non-sampling error? What do you think? Pause here if you have to. The answer is this is a sampling error because Bobber did everything correctly. However, the results we obtain from sample could be slightly different from the actual population. And in this case, the only thing that Bobber could improve, he could collect a greater sample. The sample of 10 is very small. However, otherwise everything was done correctly. Example 2. Prior to the 2016 presidential elections, Bobbert surveyed all his Facebook friends, of which 7 said they would vote for Trump and 43 said they would vote for Clinton, and 36 did not respond. The actual population vote, as I previously mentioned, was 46.1 for Trump and 48.2 for Clinton. So is this a sampling or non-sampling error? And I hope that you see that it is a non-sampling error. It's due to poor design. First of all, because Bobbert was asking his Facebook friends, he omitted a large portion of the population. He basically selected only people who use Facebook and which represent a certain type of a population. Plus, we have a high non-response rate. See, 37 people did not respond. So that's a non-sampling error as well. Okay, so in reality, a sample will never be as good as the entire population. So the best way, if we want to avoid the errors, is to survey the entire population. However, it's not always feasible and very time-consuming and costly. And since the sampling error is unavoidable, we try to minimize or eliminate the sampling errors. So let's talk about the golden standard. So in order to minimize or the sampling error, one important thing is that our sample was a random sample. So we have to come up with techniques for randomization of the way we select subjects for sample. Also, it has to be blinded, and it has to be like if we're trying to prove causality, not just show association, it has to be a cohort study. Cohort is another name for group. So we have two or more groups that we apply different treatments to, and we're trying to, and we later we're going to compare the results in those groups. So here's an example of one of the biggest experiments that was done in the health industry. In 1953, an experiment was designed to test the effectiveness of the Salk vaccine in preventing polio disease. Polio was very prevalent, and as a result, a lot of children died or became paralyzed. So 400,000 children were selected, and then those 400,000 people were divided into two cohorts. Each one consisted of 200,000 people, and so the selection into groups had to be random, like by a flip of a coin. So, and then after that, one group was um, given the actual vaccine, and another one was given a fake treatment, like a sugar pill. Well, since it's a vaccine, I think it was a saline solution at that time. So this administration of the vaccine or the treatment was blinded, meaning the people who administered the vaccine and the placebo, they did not themselves know which group the person belonged to. So this is another very important aspect of a randomized study. And then they compared the results from two groups. And in the treatment group, only 33 developed paralytic polio. And in the placebo group, 115 developed polio. So thus, the conclusion was made that the vaccine actually works. It is effective. So here's some vocabulary that you need to be aware of. A treatment is a specific condition or drug applied to the individuals in an experiment. So in the previous example, it was the vaccine. The experimental group is the group that is exposed to the treatment. So that's the group that actually received the vaccine. The control group is the group that does not receive an experimental treatment. So that's the group that gets a placebo. And that's the group that we're comparing the results with. And placebo is a dummy treatment, and sometimes it's a sugar pill, sometimes it's distilled water or saline solution. Why do we need a control group? Because to eliminate the a placebo effect. A placebo effect when a person's health or mental uh, situation improves just because they were given something, even if it's a sugar pill. This effect is widely known. Some more vocabulary terms. We have the explanatory or independent variable that are thought to influence the response or outcome or dependent variable that we want to study. So in our example with the Salk vaccine, the actual vaccine was independent variable. That was our explanatory variable. And the result, whether the person got polio disease or did not, that was a response or an outcome variable, which depended on the explanatory variable. Then another term is blinding. And blinding in an experiment is desirable to keep the information about the treatment hidden from the patients and anyone involved with evaluating the patient. Because again, to avoid the placebo effect, it is called double blinding if both patients and evaluators are unaware whether or not the patient was given a placebo or an actual treatment. And it's a single blinding if only patient is unaware. Double blinding is considered a golden standard, better, because sometimes even evaluators could have a placebo effect themselves. So they may think that because they know what's going on, who got what, that also may influence their evaluation. So it's best when both the patients and the evaluator do not know. So blinding prevents conscious or subconscious biases or expectations from influencing the outcomes of the study. All right, in the next video, we're going to be talking about organizing statistical studies.